Please, somebody let me know if we're okay, going live. And so just for me to know, the decision is that I will not stop the live streaming when I leave. Allow recording. Okay, I've approved of your recording, David. I'm gonna make you a host like we did before. And with that, we're getting very ready to be ready. Welcome to those of you joining us. We are I'll give people one more minute and then we're going to get started. Okay, I think we can get started. Thank you for joining us to another exciting, fine seminar. Last week we had what most of us thought it was a fantastic, terrific panel with editors of behavioral sciences journals. I heard from many of you, some people who did not ask questions but were attending, and I got, hold on, I need to shut down my, who got back to me saying that they really enjoyed the panel and so did the editors. So we will continue offering different kinds of panels as we go along through the fine seminar series. I don't think we have any more panels this semester, but we already have one planned for the fall of 2023. So today, what we have, today we have Noah Snyder-Mackler, who is going to be sharing some of his research, and it's a somewhat different introduction I can make of him, one that doesn't help my realization that years go by and we're getting old, because I met Noah when he was an undergrad at the University of Pennsylvania. At the time he was beginning to do some research with cowbirds, studying parasitism. And so he had already become interested in bringing technology to the study of animal behavior. And so I remember that one of the things he suggested we tried was to do recordings of our data collection procedures in Tiputini in Ecuador, where Tony Di Fiore and myself, we have been working for a few years on a number of peer living taxa down there. So Noah went to Tiputini, went through the tropical forests of Ecuador, and he completed his undergraduate degree, and after that, and he studied his PhD, again at the University of Pennsylvania, now working under the guidance of Dorothy Cheney and Robert Seyfarth. But he did not work with the baboons that they were studying. Instead, he started working with the geladas in Ethiopia. He completed his PhD. He went on to do a postdoc fellowship at Duke University with Susan Alberts. And after that, he went to Washington, University of Washington in Seattle, where he was first a postdoc, and then he took on an assistant professorship position for a couple of years. After being at UW, both in biology and psychology, he moved to Arizona, where he's now an assistant professor in the Department of Evolutionary Anthropology. He was just telling us, those of us who joined early, and remember that we invite you to join the meeting a quarter to the hour if you want to have more of an informal conversation before the seminar gets started. So he was just telling us about his recent trip to Ethiopia. Some of you may be aware that the country went through some difficult times of civil unrest, but apparently field conditions are getting somewhat normal. So today, Noah will be, I want to be sure that I cite properly the title of the talk. If you bear with me, I know he's been doing a lot of research. Okay, there we go. Thank you, Noah. He'll be talking about social and environmental modifiers of immunity and aging in non-human primates. Like we're always doing fine. I ask you when we finish, I need to go. I've already explained this to Noah, so I'm not going to be around during the presentation nor during the questions and answers, but David and Alba have very kindly volunteered to do it. And so you know that you'll just type question marks to ask questions, and I'm sure we have another round of wonderful debate and discussion around the research that Noah has been doing now for, we're getting close to what, 10 years since he finished his PhD. So Noah, thank you very much for joining us today. Thank you all for attending another fine seminar. And Noah, you can take it from here.
Awesome. Thank you so much, Eduardo and Alba, for the invitation, and Eduardo for that really nice introduction that also makes me feel a little old now. Going back to 2004, 2005, when we first got to know each other, and back to my days as a budding ornithologist, and then thinking about how, you know, these arboreal primates are really just, you know, very similar to the birds, and so we can use some of the same data collection techniques on them. I don't know how that ended up working out in the long term, but it was a really great, wonderful field experience for me. And helped sort of solidify that this was the career I wanted to choose moving forward. All right, so today I'm going to talk about some recent work we've carried out in my lab here, showing how environmental adversity, so exposures to adversity, can impact the rates at which we age, how our bodies are aging, and ultimately, you know, disease and death. You know, the three sort of key components of Darwinian fitness are health, reproduction, and survival. Health and survival are obviously tightly linked to the aging process, as well as reproduction. And we know that reproduction is one of the strongest predictors of reproductive output, especially in female primates, is the length of their lives, so how long they live. So the talk today is roughly going to be broken up into two parts. The first is going to be about how exposure to an adversity in the form of environmental adversity impacts immunological aging. And the second shorter part about the social environment, how the social environment plays a role in mitigating some of these adversity associated costs in non-human primates. And I know I saw on the schedule that Lauren Brent is going to be giving a talk in just a few weeks, maybe towards the end of this month. Lauren and I collaborate very closely studying this population of rhesus macaques in Puerto Rico on Cayo Santiago, a photo of them here. She leads much of the behavioral, oversees all of the behavioral data collection and analysis, and we work closely. So I'm going to try and not to step on her toes too much in terms of the work that she'll be presenting. So I might just tease it a little bit. Okay, so from what I understand, the core theme of this seminar, this fine seminar, is to understand how and why animals live in different societies, right? Here, I'm going to talk about the consequences of some of these societies and what that means for animal health and well-being. Now, work in my research group here is motivated by the strong relationship between the environment and health across species. You know, here is just a schematic from a review paper that I sent around to all of you as one of the readings for today's seminar, depicting just sort of a schematic of the link between the social environment, here spanning sort of social adversity on the left to social advantage on the right, on health and survival across a number of different social species. Now, the overarching goal of the work is to gain a better understanding of the health consequences of variation in the environment, both the social environment and the physical environment, how organisms navigate their environments and those consequences, and how their navigation and experiences impact health, reproduction, and survival. And ultimately, some of the work that I do also aims to understand how this work might help us better understand and potentially treat human health and disease. And so I'm going to spend some time talking about a species that may not have been discussed as much in this seminar throughout the past year, and that's us humans, right? These the primates that we are, and for humans, for us, one extremely salient component of our environments is our social and built environment. We know these as the social determinants of health. These are the environmental factors that pattern health in our society. And they include things that come from education, access to access and quality of that education, to our neighborhood and built environments, economic stability, access to healthcare, and the social and community context, sort of our social embeddedness. And these social factors all really come together to form our lived environment, our lived experience, and impact many aspects of our lives. For instance, here's a figure from a paper that came out in 2016 showing life expectancy in the United States, data collected from 1.4 billion de-identified tax records collected between 1999 and 2014. So if you were alive between 1999 and 2014, and you paid your taxes, you are part of this data set. And what we can see is that there's a really clear socioeconomic gradient in health, so that the income patterns health. This is completely unsurprising to us. People at the higher end of the socioeconomic spectrum live longer than those at the bottom. Now, this discrepancy is about 10 years in women and 15 years in men. And when I'm giving this talk to people at the National Institutes of Health and things like that, I try to put this disparity in perspective. If we were to cure cancer right now, right, that would lead to an increase of an average life expectancy of about 2 to 3 years, right, overall. So this income discrepancy, this social disparity in survival is enormous. And it's largely been getting bigger over time, sadly. And here's just showing those at some of the higher ends of the income bracket making $250,000 a year, and those at the very low end making $17,000 a year. Men on the left, women on the right. And what you can see is that life expectancy, while it's increasing across the board, this increase is increasing disproportionately. The wealthy are benefiting more, and this is leading to an increase in this mortality gap between the lower and the higher ends of the income spectrum. And these social determinants of our health and well-being, right, are not just linked to survival outcomes. Lower income individuals, lower social status individuals, let's say, also have higher rates of many physical diseases, many mental health diseases, and overall rate themselves as being in poorer health. Now, given our shared biology and evolutionary history, again, I mentioned at the beginning, we're primates as well, it should really come as no surprise that many non-human social animals also experiences these same social and environmental challenges. And as a consequence, they may also, and do also, exhibit similar health and survival gradients. Now, this is because while our current environments and disparities aren't necessarily natural, they're built, they might be features of some of the environments in which we currently live. Our responses to adversity and the unpredictability that accompanies it is very natural to us and is deeply rooted in our evolutionary history. For instance, and this also comes from that review paper that I sent around, lower social status animals from mice all the way up to monkeys show signatures of poor health and have shorter lifespans than their higher status counterparts. Now, we pulled this list together, this review paper back in 2020, and likely this list was completed in about 2019. And I know that many more studies have been added to this in the almost 4 to 5 years since. So these observations raise a really natural question, which is the focus of work in my group, which is what are the biological processes that account for some of these strong associations between adversity, health, and survival? This is a really interesting question from an evolutionary perspective to try and understand what are the causes and consequences of variation in reproductive fitness, but also from a human health perspective, this is relevant to improving disease prediction intervention, understanding the causes and consequences of social inequality, and putatively its relevance to human health. And so a lot of the work in my group is really trying to understand how does adversity, particularly social adversity, get under the skin in sort of a colloquial manner, to impact health, reproduction, and survival. Moreover, how do organisms navigate their environments to mitigate or even potentially offset some of these exposures to adversity? So in my lab, we're really trying to get at some of these cellular and molecular changes or consequences to exposures to different forms of adversity, from the social to the nutritional, which I'm not going to be talking about today, as well as to the physical, so the climatological changes that occur. So one thing we know is that across many social organisms, social status patterns are access to a number of resources. And it's not necessarily always just the access to resources that impacts our health, reproduction, and survival. But what one of the hypotheses about why low status might be particularly detrimental to health, reproduction, and survival is that those animals that have maybe more social support, less social support, or are lower status, because they have less predictable access to resources, they may have a more unpredictable environment. In other words, they don't necessarily know what's happening next for them all the time, right? They don't have as predictable of a future. I think as many of us might be familiar with, if we don't know what's coming next, we might be a little stressed out, right? And so some of the ideas is that some of the ways in which our environment sort of become biologically embedded, right, into our bodies and impact our health and well-being is through sort of this activation of our chronic stress response. And so this cascade, this chronic stress response can have sort of cascading effects on many components of our bodies and our physiology, and ultimately impact sort of different aspects of our immune system and ultimately health and survival. And so many of these changes that occur in response to, let's say, the chronic stress in the face of adversity are also some of the changes that are also sort of outcomes that we see in individuals as they age. So some hints about the underlying mechanisms of how our environments, become biologically embedded and alter our cells and ultimately our health and well-being lie in the fact that exposures to these adversities, here showing measures of different social adversity, also lead to increases in the incidence of diseases of aging. So many diseases that increase in frequency in humans as we get older, from heart disease, lung disease, to strokes, and many other sorts of these diseases. So this sort of leads to the hypothesis, or just the, posits that exposures to these adversities, may accelerate biological aging of individuals, right? Leading us to, poorer health and closer to, sadly, death. In fact, I think this is, this idea that psychological stress or chronic stress can accelerate aging, can visibly accelerate aging, is actually something we're probably very familiar with, right? Many of us, think of things like hair graying, right? We see people who may be under particularly high amounts of stress that it may mean that they are sort of aging before your own eyes. And recently, Martin Picard at Columbia showed that we could actually quantify the stress-induced hair graying, which was just shown through that GIF that went through one of these hair follicles, showing in quantifying it happening on relatively short timescales here. Even so here, chronic stress, but chronic stress over a few months, not chronic stress over many years. They developed this new way to digitize and measure small changes in color along single human hairs. And the stress, the somewhat dark, but also kind of funny aspect of this is that the stress was during a grant submission deadline. So you can see sort of the graying happening around about, in December, and maintaining, white hair and gray hair up until the grant was submitted in mid-February or in early March, and then it reversed again. So again, something maybe many of us are familiar with, a bit too familiar with maybe. But some of these stress-induced changes, some of the questions that come out of this are these stress-induced changes sort of recapitulating the effects of aging, not only at the way we sort of look, but what's happening at the molecular level, and in our immune cells in particular. So does exposure to adversity or stress accelerate the aging process? And these are interesting questions from, again, a basic evolutionary biology perspective to understand what are some of the consequences of chronic stress and our social environment or adversities for Darwinian fitness in many organisms, and also from the perspective of understanding human health and well-being, and maybe identifying vulnerable populations and or designing effective interventions. Now, it's really hard for us to study this link between, adversity and health and survival in humans for a number of reasons, right? One of which is that much of the work in humans, we're not really good. We're actually really terrible model organisms. So one of the things is that much of what we know from humans comes from these correlational studies. So we still don't know if a poor environment, directly leads to poorer health. Or is it the case that, as been shown in some human studies, are there potentially mediating effects, such as access to resources? So those who are under more adverse environments have less access to things like basic healthcare, right? Which has been shown to have sort of strong contributing factors in the humans. Additionally, poor health could also be due to an individual, could also lead to someone being in a poor social environment, which we call reverse causality or health selection, right? And the uncertainty of understanding the relative contributions of these two different pathways, and also this direct pathway down the middle, is compounded by the absence of information about social and biological conditions at the beginning of many studies, so prior to start dates of key studies, as well as the consistency of these social environments over time. And to control for these possibilities, we can actually turn to animal models, some of them shown here, where we're able to eliminate variation in health behaviors in wild population in particular. And then in experimental settings, we can assess sort of the direct and causal nature of this link by eliminating some of the reverse causality as well. So the first part here, I'm going to tell you about a study I did during my postdoc with Jenny Tung when I was at Duke. And then we'll shift to some of the more recent work we've been doing on Caio Santiago and in the face of another form of adversity. So in this first study, it was involved in some work using captive female rhesus macaques as a model to understand how social adversity can causally alter immune function. These rhesus macaques are a good model for human social hierarchies because they also form structured hierarchical societies. As in humans, these hierarchies impact macaque health and survival, both in captivity and in more free-ranging naturalistic populations. From a biological perspective, they share many core neural and genetic pathways with humans. They're primates like us. And in this captive population, we're able to really experimentally manipulate social status, which we quantify as dominance rank, which all of you are very familiar with, which allowed us to test for the causal and direct effects of social status on immune function. So to do this, and I'll just briefly go through this, we use this paradigm where we introduce females into new social groups and where the order of introduction into these groups strongly predicted subsequent dominance rank. So we took females who were totally unrelated, no prior social history with one another, and introduced them into new groups of 5. So this graphic here depicts how females who were introduced earlier into these groups had a kind of home field advantage and attained higher dominance ranks in their social groups. So the female who was introduced first became alpha, the female was introduced second, beta, and so on in general. And we created 9 replicate groups here. Again, this work was led by Jenny and her colleagues, Luis Barrero and Mark Wilson at Emory University. Here's a sort of graphical representation of the formation of one of our study groups. On the x-axis, we have time, and on the y-axis, we have our continuous measure of dominance rank, which is ELO rating, again, something that's become very common to use in terms of quantifying dominance rank in behavioral ecological research. So higher on the y-axis is higher dominance rank, and lower is lower social status dominance rank. Each line's one monkey, and they're colored based on the order in which they were introduced into the group. Okay, and each point represents a change in dominance rank for that female because she engaged in some sort of dominance interaction that we observed through regular behavioral observations. And just to put a point on it, these dominance interactions were largely non-contact mildly aggressive sort of displacement-based interaction. So it wasn't just that these low status individuals were constantly getting beaten up and had lots of injuries. That was actually not the case. So what you can see from this is that females in this group quickly established a strong linear dominance hierarchy that remained stable for the duration of the study. So after establishing these groups, we collected blood samples from these animals and measured some of the molecular changes that were happening in their immune cells, in their primary immune cells, to try and understand how this period of chronic stress from the adversity of being low social status for over a year of their lives impacted their immune systems and might impact their health and well-being. And what we found was that those animals that were of lower dominance rank had dramatic molecular changes, many of which were involved in inflammation. So they had increases in the expression of genes that were predominantly involved in the inflammatory pathway, right? And when we have chronic elevated inflammation, that can lead to lots of collateral damage. Our inflammatory response is actually really good in the short term, but it's also pretty non-specific. But when chronically activated, it can lead to a lot of chronic collateral damage, damaging our tissues and our cells. And that's what we see as sort of many of the diseases that we see with aging, many of these non-communicable diseases like cardiovascular diseases and such are due to sort of this runaway chronic inflammation. So we saw this increase in baseline sort of levels of inflammation in these females who were of lower social status. And we also found that upon stimulation, so we took some of these cells from these females and we stimulated them with different infections, in vitro, and we found that not only were low status females, not only did they have higher baseline levels of inflammation, but they also exhibited a stronger inflammatory response to an infection, which suggests that there was this exaggerated inflammatory response. So not only did they have lots of potentially collateral damage at baseline, but also when there was an infection, they may have been overreacting or over-responding to that. So we then wanted to molecularly test one of the primary hypotheses underlying why chronic adversity might alter immune function, and that is through chronic activation of the stress response pathway, which we lead to secretion of glucocorticoids, what we consider our canonically our primary stress hormones, and when chronically activated, there's a negative feedback loop where when acutely activated, our glucocorticoid response pathway actually turns itself off. But when chronically activated, there becomes that off switch becomes desensitized, right? And we call this glucocorticoid resistance. And when glucocorticoid resistance happens, one of the primary things that glucocorticoids also do is they act as really potent anti-inflammatories. So if we think about that, we're not able to turn off this feedback loop anymore, and we have chronic elevation, we actually might see runaway inflammation. And so we did some molecular work trying to look and see, how the glucocorticoid binds the glucocorticoid receptor in these animals in their immune cells, and what that might mean for turning on and turning off gene expression, particularly in response to the inflammatory response. So what we did is we took the cells out of these animals and we conducted an in vitro glucocorticoid stimulation, which means that we gave them a synthetic glucocorticoid. These cells, treated them for 90 minutes, and then we wanted to see how that altered, not only where the glucocorticoid receptor is binding in the genome, so how active it might have been in low status versus high status animals, but also sort of what that means for the females who are lower status in terms of what genes were turned on and turned off. And I just need to hop to my slide navigator here. And what we found was that, we did a lot of sort of bioinformatics looking to see where this glucocorticoid receptor might be binding in the genome in lower status females versus higher status females in response to this treatment with a synthetic glucocorticoid. And I've got to show you sort of canonically for a transcription factor to bind and to affect transcription, the region where it binds needs to be accessible or open, which means the chromatin, these little, gray circles, can't be tightly packed. So we looked at the binding sites for the glucocorticoid receptor we found that in low-ranking individuals, it was a lot less accessible, meaning that they can't really bind, right? However, in these high-ranking individuals, it was more accessible, and the glucocorticoid receptor could bind to the site. And so what we saw is that this glucocorticoid receptor was able to bind at this site and turn off inflammation more readily in the high-status individuals compared to the low-status individuals, sort of providing some evidence for the molecular evidence for embedding biological embedding in the hypothesis that the chronic stress of being low status led to glucocorticoid resistance at the molecular level, so it might be driving this inflammatory response. So these, I started briefly talking about aging, and now I'm going to sort of circle back a little bit. So do these experimentally induced changes also accelerate the aging process? And so during my postdoc, we weren't able to look at this within the same system, but we did compare our experimentally induced social adversity to human aging across macaques to humans. We found really broad similarities between aging and social adversity in how immune cells were turning on and turning off different genes. So as I mentioned in those animals in the experiment we just showed you before, those who were lower status, purportedly faced more adversity, were more chronically stressed, had higher levels of inflammation, but also overall they had immune profiles that looked older for their age, right? So they had immune profiles that looked more similar to older individuals, older humans, than to younger humans. But this study was done sort of across pathways and across species and across different studies, and was really a first pass at this. But a lot of this work during my postdoc really set me on my current trajectory. Like, I'm excited about identifying the molecular consequences of adversity and aging, and how we can harness this knowledge to understand how animals can navigate their environment and respond to their environments, and also how we might be able to use this to understand and improve human health. So one of the first projects that we carried out in my lab was examining the effect of one extreme form of adversity, a natural disaster, on organismal health, aging, and also behavior. Now, much of this work here revolves around rhesus monkeys that live off the coast of Puerto Rico on this island here, Cayo Santiago. As many of you know, macaques are not native to Puerto Rico. They're native to Asia. This research colony was founded in 1938 when C.R. Carpenter brought back 409 rhesus macaques and plopped them from India onto this island. Today, there are about 1,800 animals on the island. It's a 38 acre island, so not very big. And these animals are the direct descendants of the founding population, and they're managed by the Caribbean Primate Research Center and the University of Puerto Rico. So they are a research center funded by the National Institutes of Health, but they are not themselves a national primate research center, of which there are only a handful. Overall, the animals are provisioned with food, but otherwise they're left to their own devices, apart from once a year where they can be tracked to obtain biological samples. So this allows us to collect some really detailed longitudinal data and represents what we think is a phenomenal sort of natural laboratory, a nice hybrid between sort of really captive lab settings and fully wild populations. And this allows us to leverage some of those tools and access that are available to the captive studies, combined with the naturalistic and ecologically relevant complex social environment that these animals form in the wild. Now, in September 2017, a Category 4 hurricane made a direct hit on the island of Puerto Rico, devastating the island and leaving massive damage in its wake. I just got back from Puerto Rico last week, and much of the island, including the people and the infrastructure, are very much still recovering. Just over 5 years later. This was actually compounded by another hurricane that hit just last year, Hurricane Fiona, which was a much weaker hurricane, but because of the substantially weakened infrastructure from subsequent to Hurricane Maria, Fiona still inflicted a lot of damage on the island. This hurricane in 2017 also passed directly over Cayo Santiago, which you can see here in this Doppler image, sort of zoomed in, and in the inset, there's Punta Santiago, and then Cayo Santiago is that little blip just southeast of that star. So in the eye of the hurricane. So it's sort of the Hurricane Maria came in, as category 4 and hit Cayo Santiago and the southeast coast of Puerto Rico first. So there was massive damage to the island in terms of there was limited infrastructure on the island for supporting the colony, but for the most part, there were lots of trees and foliage and things like that. But there was massive destruction of all of that on the island, Cayo Santiago Island. But despite this really extreme event, mortality was really limited. Less than 2.5% of the population died in the immediate aftermath. And actually, there was no differential survival based on age or sex in the year after the storm compared to prior years. And this 2.5% number is in line with other work showing really no overall increases in mortality after some of the other previous major hurricanes that hit the island, Hurricane Hugo in 1989 and Georges in 1998. And this slight blip in mortality is actually not statistically different than a 3-month stretch in any prior year, even outside of hurricanes. So it wasn't that there was just this huge increase in mortality. This provided us with an interesting natural experiment to try and identify what are some of the consequences of this extremely adverse event where the animals had to stay on the island and had to live on the island through this event, and then in the aftermath of the event in this changed environment for multiple years. And in humans, many individuals, there's sort of selective immigration after these events. So that's another one of the challenges to try and understand what are some of the consequences of exposure to extreme natural disasters on human health and well-being, is the fact that some individuals are able to leave that either before or just after the storm. But here, these animals had to remain on this island, and they all have the same access to the same sort of resource support, nutritional support, and things like that after the hurricane. So we know that survivors of extreme natural disasters and adverse events like these hurricanes have increased incidence of many diseases of aging, like cardiovascular disease and also chronic low-grade inflammation. So the question we set out to ask here was, how does a natural disaster increase the incidence of some of these diseases of aging? What are some of the molecular consequences? And does it actually accelerate the aging process in some of these individuals? And then towards the end of this talk, I'm going to say, what are some of the ways in which these animals maybe mitigated some of these effects, or what predicted who was more vulnerable or resilient to this environmental stressor? So my former PhD student who just defended about 6 months ago, Dr. Marina Wadowicz, drew on our historical sample collection from 2014 to 2016, as well as prospective data collected after the hurricane from about 550 animals. This is an opportunistic cross-sectional study, and the pre and post-hurricane samples were roughly matched by age and sex. There were actually no statistical differences between the ages and sexes of animals sampled before and after the hurricane. The goal here was to test how the hurricane molecularly affected the immune cells in the monkeys on Cayo Santiago. So what Marina did is she generated RNA sequencing data, which here is just trying to understand all of the genes that are expressed in these immune cells and seeing if any genes are differentially expressed, indicating differences in protein changes later on and differences in function in those immune cells between individuals sampled before and after Hurricane Maria. And here we were looking specifically at blood samples, so non-destructive sampling of these animals, which contain our leukocytes, our white blood cells, and some of our primary sort of inflammatory responders. So first, Marina found that Hurricane Maria significantly altered the expression of about 4 to 5% of the immune cell expressed genes. So in this work, we see sort of the first molecular signature of this extreme natural disaster on sort of the molecular changes in our immune cells for the first time. As expected, we also saw age-associated differences in immune cell gene expression. About 15% of the genes that are expressed in your blood change as you age, or are more highly expressed in older individuals than younger individuals, or vice versa. So the next natural question we asked, and given the setup to where I've come forward in this talk, was, are these age-associated genes also associated with exposure to the hurricane, right? So are these genes that are differentially expressed between older and younger individuals also differentially expressed between individuals sampled before and after the hurricane? And what this really phenomenal animation of a Venn diagram is showing you is that, interestingly, twice as many of the genes than were expected by chance were significantly associated with both the hurricane and aging. So this suggests that there's substantial overlap in terms of the genes that are differentially expressed between older and younger individuals. Here, we measured age quantitatively, we modeled it quantitatively, but I'm just talking about older versus younger for ease of interpretation. So genes that were differentially expressed with aging are also more likely to be differentially expressed between animals sampled before and after the hurricane than would be expected by chance, suggesting that the hurricane is tapping into or affecting these same molecular pathways that change with age. We also looked at that in a directional manner as well. And so for the next few figures, I'm going to be showing you the effect sizes of age and the hurricane. So this is basically going to be the slope or the beta, also the effect size of either age or the hurricane on a given gene's expression. And in all of these plots, a given gene is going to be represented by 1 point. Positive effects means higher expression after the hurricane or in older monkeys, depending on which effect size we're looking at. And I'm going to have these little icons up at the top there next to the slides to help sort of ease the interpretation as we move forward. So the first thing we noted is when we were looking at this sort of directionally to see if, exposure to a hurricane was akin to aging a little bit more, is that we looked at the level of specific blood types that are found in your immune cells. Here we've got your granulocytes, your monocytes, natural killer cells, T cells, and B cells. So these are our primary immune cells. Some of them are more involved in our, primary inflammatory response, and others are involved more in our adaptive, more long-term learned immune response. So at the level of specific blood types, we focused on genes that were sort of markers for different circulating immune cells. Again, the reason we did this is because we were looking at whole blood when we measured the gene expression. So we couldn't tell if there were changes in different cell types with age or with exposure to the hurricane. So we looked at genes that are sort of indicative of different cell types. So genes that might be more highly expressed in CD14 positive monocytes, right? And so what we saw when we looked at these marker genes within all of these different cell types is that we saw largely similar changes in the expression of those genes in aging and exposure to the hurricane. So we see here is that individuals sampled after the hurricane looked more similar to older individuals, at least at the level of these genes that are markers for some of our primary immune cells. Now, when we expanded to look across sort of all of the genes that we measured, that we quantified in the blood cell in this heterogeneous population of white blood cells in the whole blood that we sampled, we saw that there was a weak but significant and positive correlation between the effect of hurricane on a given gene's expression and age on a given gene's expression on the y-axis. So each of the points here represents a single gene that we measured. The x-axis is the effect size of the hurricane. The y-axis is the effect size of age on that gene's expression. So overall, there's a significant positive correlation between the two effect sizes, such that on average, older age and post-hurricane sampled individuals exerted similar effects on a gene's expression. For instance, I'll give you one gene here, which is called Veriscan. The shorthand for that is VCAN. It's a pro-inflammatory protein that shows significantly higher expression in older monkeys and after the hurricane. And I hinted at pro-inflammatory and pulled out our pro-inflammatory gene for a particular reason, given some of the work that I showed before. So not only can we look at sort of the correlation of this of effect sizes of hurricane and age on a given gene across genes within this sample, we can also look at we may not expect the effect sizes are going to be exactly correlated, right? So that the direction and magnitude might not always be perfectly correlated or really tightly correlated, but the direction of these effects will be concordant. such that genes that are more highly expressed after the hurricane are also more highly expressed in older individuals and vice versa. And when we look at this and we break them down into genes that are significant with or differentially expressed before and after the hurricane and differentially expressed with age, we can see that 83% of these genes are up in the top right or the bottom left quadrant here, which is significantly more than we would expect by chance. We would expect if there was no association between the effect of the hurricane and age, they would be in all four quadrants pretty much equally. And that's certainly not the case here, suggesting that exposure to Hurricane Maria recapitulates some of the effect of aging on immune cells. And then we can look within each of these different quadrants and say, okay, what are these genes possibly doing, right? And what does that mean for the organism's, immune function and putative health and well-being? When we look at these genes in the top right, we see that many of these genes that are increasing, they're more highly expressed after the hurricane and in older individuals are involved in the inflammatory response, suggesting that this exposure to this adversity increases the inflammatory response in a similar way to the aging process. Now, in the bottom left, we see that many of what we call these sort of hallmarks of aging, telomeres might be something that many of you are familiar with, are decreasing in response to the hurricane. So we see that after the hurricane, there's a downregulation or a decrease in the expression of genes that are involved in telomere maintenance, the DNA repair response, which we know goes awry as we get older, and also B cell proliferation. We know that as we get older, our B cells, these are sort of our primary adaptive or learned immune cells, decline with age. I think I'm going to skip this slide due to time. That's okay. We saw similar things in the protease-based network. So the next question we asked is we want to see if exposure to a natural disaster actually accelerated a global measure of immunological aging here. So on average, you and I, all of us on this call right now, our body's aging at the same rate as our chronological age. So if I'm 22 years old, which I'm not, but if I was 22 years old, or maybe when I met Eduardo when I was 22 years old, ideally the biological age of many of my organs and my immune cells was also 22, right? Here we've got this monkey, depicted on this X equals Y line, where they are 15 years, they're chronologically 15 years old, and also we measure their blood and they are biologically 15 years old, or we can quantify that in some manner. But this is not really always the case, right? With some individuals being biologically older than their chronological age and others being biologically younger than their chronological age. We can think of these as sort of like different biomarkers for aging relative to your chronological age. And I mentioned telomeres on the previous slide, and telomeres are one biomarker of aging that might, capture acceleration or deceleration of the aging process in response to different environmental exposures or experimental interventions. So we wanted to test if the hurricane increased relative biological age. And to do so, we drew on this transcriptional aging clock. So this aging clock based on gene expression data that was developed in humans back in 2015. Developed in humans, different species, primates as well, about 94% shared DNA sequence with rhesus macaques. And you can think of this transcriptional clock that we used as an analog to the well-known epigenetic or DNA methylation clock that many of you may be familiar with, but that is really strongly predictive of chronological age. And then the difference between your biological age and your chronological age is predictive of health and mortality as well. And so when we deployed this, we actually found that monkeys that were exposed to the hurricane appeared approximately 2 years older than those that did not experience the hurricane. So to put this in and phrase this another way, if we sampled an 8-year-old monkey prior to Hurricane Maria, they had sort of the transcriptional age, or their blood looked about the age of an 8-year-old monkey. If we sampled an 8-year-old monkey after the hurricane, their blood on average looked like the blood of a 10-year-old monkey. So they accelerated their age about 2 years. And we can do some sort of like back of the envelope math here and translate that to human lifespan and say that their lifespan is the rhesus macaque lifespan is about 3 to 4 times faster or more accelerated than the human lifespan. So if we multiply this by 3 to 4, this change, this age acceleration of about 2 years is similar to about 7 to 8 year age acceleration in the human lifespan. So relative to the rhesus macaque lifespan, this is a lot. This is a big effect. Dr. Cassandra Turcotte, a postdoc who was working on this project, also found that the hurricane was not only associated with sort of the molecular aging in the immune cells, but also with some broader phenotypic changes, other aspects of the aging process, like our mobility. So individuals sampled after the hurricane had reduced mobility for a given chronological age. And so we're now sort of expanding this out into other domains and phenotypes of aging, not just looking at the immunological process and even looking across different tissues and organ systems in animals that we're able to sample. But the sort of the more powerful implication of these approaches is that we can now look at sort of population-wide variation in multiple aging phenotypes relative to chronological ages and quantify heterogeneity in the response to these stressors. This increased aging phenotype was not really the same across all individuals and all bodily systems, right? So I gave the example of that 8-year-old monkey on average, that average 8-year-old monkey that on average looked like it was 10-year-olds after the hurricane. But some 8-year-old monkeys sampled after the hurricane looked like they were 8 years old. Others looked like they were 10 years old, 12 years old. Some looked like they were, Benjamin Button-y and maybe 6 years old. So some of these animals didn't exhibit this accelerated aging phenotype, while others did. And the question is why? And this is the question we're really diving into at the moment. And we think it lies within that social environmental axis we know plays a big part in an organism's health and survival that I mentioned earlier. And again, we can think about this because of its well-established link to health and survival across species. And particularly when we talk about sort of the stress response pathway and unpredictability, there is evidence supporting how being in a more positive social environment or having more social support might be protective in the face of adversity. This is often called the social buffering hypothesis. And again, as an aside, this points to another powerful aspect of sort of studying non-human primates or other non-human animals, because we can objectively quantify variation in the social environment, which Lauren's going to be telling you a lot about in just a few weeks in the seminar series. We actually found that, in work led by Kamita Starr, a PhD student at UPenn, in work led by Lauren Brent and Michael Platt, that the hurricane had pretty strong effects on social behavior of these macaques. We found that these animals after the hurricane built new social connections. But this wasn't that everyone built new social connections, right? On average, many did, but the animals that were the most socially disconnected or most socially isolated before the hurricane actually exhibited the greatest increase in their social connections. Perhaps, this reflects some sort of buffering strategy for those that were more socially isolated to begin with in the face of this really traumatic and intense environmental stressor. So we're currently looking into how these social factors like social connectedness might predict heterogeneity in the molecular, physiological, immunological response to the hurricane, which is work that we're starting to dig into, as we speak in this early project. So Lauren Brent, who's going to talk again here in a few weeks, is actually going to tell you, I hope, about some really exciting new results that Camilla's led with Lauren that'll tell you, showing sort of that the impact of social connectedness on mortality in the years after the hurricane, now looking at about 5+ years out. So just to sort of sum up a little bit here, the impact of the social environment on health and aging is the focus of a large collaborative project that Lauren, James, and Mike Montague, many of you know, and I are leading. We're in year 4 of this study, and so we're looking to see how variation in the individual's physical and social environments impacts different components of the aging process, and how that relates to their health, reproduction, and survival. So some of the earlier work here is led by a graduate student of mine, Mitchell Sanchez-Rosado, who found that some immune cell types are actually differentially change in their function, change in their abundance with age, and that is dependent upon their social status. Here showing that this one cell type, these CD4 T regulatory cells, which accumulate with age and are impacted in inflammation, are actually accelerated in terms of their aging phenotype in individuals that are lower social status. So we're sort of extending this a little bit more as we speak. And then, let's see. We're also really interested in understanding what we can do about this in response to the hurricane. So Lauren will touch on some of Camille's new findings. Not only to quantify sort of the acute effects of this hurricane, natural disaster, but also look at the long-term consequences and what predicts who will be resilient or more vulnerable to this, and also what the trajectories look like. So how animals sort of navigate this new environment and use their adept social skills to maybe protect their bodies for a little bit longer. And so with that, I just want to say sort of that this work sort of shows jointly that the adverse environments really do induce molecular changes that impact our immune cells, and in some cases accelerate the rate at which our bodies age. And when combined with the work that we're doing on understanding social environmental modifiers, we're starting to identify the mechanisms that might underlie how adverse environments impact healthy reproduction and survival, and what animals can do about it to protect themselves and live healthier, longer lives. And so I just want to put a flag here that, it's a little bit over 100 degrees right now here, but it's still very nice and beautiful, and lots of desert fauna out there, flora and fauna. So come join us. We're always looking for postdocs and PhD students down here at Arizona State University in my lab, where we're trying to integrate sort of the outside and the inside worlds to get a better understanding of animal and human evolution, health, and well-being. And I also want to thank all of the close collaborators here, particularly Lauren and James and Michael and Mike, Melwin and Carlos, who run the Caribbean Primate Research Center, and then Marina and Camille, who led a lot of the work that I talked about. And then finally, this phenomenal crew of researchers studying humans and non-human animals to try and understand the social determinants of health and survival, which really form the foundation for a lot of the work that I've been carrying out in my relatively new lab. And with that, I'll take any questions you might have, and we can sit around and chat.
Thank you, Dr. Snyder-Mackler. If anyone has questions, you can post a question mark in the chat, and then we will run through that way. All right, Clara, would you like to open things up? Oh, I think you're muted.
Yeah. Clara B. Jones, retired social evolution. Thank you for this talk. It's just, in my opinion, cutting edge work, and I really commend you. I have an observation, which is an elementary one, because I don't know your literature. And then I have a question, both very short. First of all, the observation about the natural experiment, which is just beautiful, but hurricanes are density independent events, and most mortality in the field is density dependent. So I'm wondering whether or not there could be comparisons with density independent and density dependent mortality or effects.
That's a great question. I don't think we can get at that on the Cayo Santiago Island because it's so small and it is pretty dense and densely populated as well. I might want to flip that question. We have a lot of time here to chat, right? I think generally, yeah. All right, so I'm going to flip that question, maybe turn it into a discussion here a little bit. Clara, because I think it's really interesting, is what do you think it is about the about dense populations that might make the impact of a hurricane or a natural disaster like this more impactful? I mean, in my mind, I think it would be that there'd be more limited access, there'd be resource scarcity, right? After the fact. Do you have any other sort of thoughts on what might be driving some of those things?
Well, you have a random, thinking of it abstractly in statistical terms, you have with density independent mortality an event. You have random mortality, and with density dependent effects or mortality, you have non-random effects. A lot of what you are calling, in my opinion, as I think of it off the top of my head, a lot of what you are calling quote unquote social outcomes might show changes from a random event to a non-random event.
I see. I think, I think some of the, I need to think about that a little bit more, because when I think about these density dependent things, I'm thinking more about the patterning of resources before and afterwards, and how they become, it becomes a resource scarce environment. And in general, we saw, the provisioning of these animals means that at least in terms of nutritional availability, it's there. But as you saw in terms of the foliage and the shade actually that's there. So the environmental resource, this physical environmental resources on this really hot Caribbean island became extremely scarce. And that's work that Lauren's going to talk to you about a little bit later this month. But that's a good question about what might our expectations be in a more density independent versus dependent manner, I guess. And then you had a second question. Yeah.
Yeah. And then I have a quick question. Are you using generative AI tools in any way to augment your research?
That's a good general question. The answer is right now, no, but that's on the horizon. We're actually using a lot of we're using different machine learning tools to try and maximize what we can get across what we see of this really interesting multimodal data that we're looking at in terms of at the organismal level, at the social level, so the population level in terms of behavioral changes, environmental changes at the organismal level in terms of different aspects of their body, and then also sort of within individual cells as we see the churning and turning of different transcriptional and regulatory machinery. But so yes and no, but maybe yes soon.
Thank you.
Yeah.
Very nice.
Thanks.
All right, next we have Carsten and then Raghavendra.
Yes, hello, my name is Carsten Schradin. I'm at a senior in Strasbourg, France, studying mammals and social behavior in South Africa in harsh environments. There was a great talk, and I like this combination of the comparative approach at the beginning with the science review, then the case study on the macaques, so one can look into detail at this, and of course the reference to humans. I'm always like to have terms very well defined, and maybe you said it at the beginning, and I tried to look it up in your science review, but I don't have full access here. Can you tell me again, what is your definition of social adversity? And what is the difference to social stress, or why do you prefer this term towards social stress? And how can it be measured and compared between species?
Yeah, how to operationalize, Carsten, is a doozy of a question. A cop-out answer would be, to paraphrase an old Supreme Court justice, I know it when I see it, but that's probably not the right way to think about this. So we think of as, I would say the difference between social adversity and social stress is that stress, we are tightly linking to the stress response pathway. And I actually generally try and avoid using that term. We think of sort of we think of that as sort of the mechanistic pathway through which all of these, effects, these downstream organismal effects occur. Adversity is what patterns that difference in the stress response pathway. So I know I'm not getting at how do you operationalize this and quantify it across species, but it's basically what social adversity or social advantage are things that pattern access to some resource. And it can be many sorts of resources. And then actually in the human literature, in the sociological literature, we say that social status itself and social adversity, social advantage is a fundamental cause, which means it patterns access to something, but that something can vary across time and space and populations and things like that. So I'm purposely sort of avoiding specifically answering your question and giving you sort of an answer of like how we actually quantify social adversity and social advantage. I will say that the three types of social adversity and social advantage that we focus on in that review, and I'm sorry you don't have access. If you're able to sign into, I can send it to you, but also if you go through PubMed through NCBI, since it was federally funded and it's been over a year since it was published, the open access version of that article should be there as well. But I will say in that article, the three ways in which the three types of adversities that we focus on are going from adverse to advantageous environments is we're looking at early childhood adversity, so exposures to early life adversities versus no exposures to early life adversities. Social connectedness, going from isolation to being socially integrated across different axes. And Lauren's going to talk about this. I keep plugging Lauren's talk, which is great. I'm excited that you guys are going to have her on in a few weeks. She'll be actually looking at some work that she did with one of her postdocs, Sam Ellis, quantifying different measures of social connectedness and integration. Is it the quality? Is it the quantity? And what does that mean differentially for health and or for survival? Again, in the macaques. So how you operationalize that social connectedness dimension can vary a lot. And then also social status, going from low social status to high social status, are the sort of the three domains of, let's say, or areas of adverse to advantageous environments that we looked at. But how you quantify that can vary certainly across space, time, and species.
So specifically in your figure, in your review, I mean, there you have it from social adversity to social advantage. Is it there related to the social integration, or what did you measure then the different species? Oh, that one's just the figure that we have. That was just a summary abstract, so that's sort of made up data, what that looks like there. But then what we have in the actual manuscript, we have phylogenetic trees, one for advantage, one for social status, and one for social connectedness. And then we show some examples of early life adversity, which has actually been characterized a lot less frequently across different species. So you're saying this here is not real data? Correct. Ah, okay. Yeah. Because I tried to figure out where these dots come from. Okay. Yeah, it's one of those graphical abstracts. Just a short follow-up question. Why do you prefer this term over social stress? Because with social adversity, you can have different measures, and social stress would be only one. Or what is the main reason that you try to avoid social stress? I try and avoid. I may be more of a product of my training and my early life academic environment in terms of avoiding using the word stress unless you can directly quantify that stress response pathway and how you measure the stress response, which is not always possible across these different organisms. And so I try and stick to using and it's also not always necessarily the case that it leads to changes in stress, but we do know that adversity can still impact downstream physiology and health outcomes. There's a really great paper by Scott Creel and many others in the early 2000s that was called "Are Subordinates Always Stressed?" And the answer is it's complicated. Okay, so you're saying social integration, social status, and changes in corticoid hormones would all be measures of social adversity. Yeah, I would say the first two are measures of social adversity, and the second one is the last one, social stress, is maybe a readout of social adversity. I think because that I see how you're saying you quantify that. Okay, so yeah, I understand. Okay, thank you. Yeah.
Were you able to access the paper? I can also make sure to send the link.
I will have it at when I'm at the university, but I'm at home on Tuesdays in home office, and I didn't have access. And yeah, I can access it when I'm in the lab.
Okay.
All right, next is Raghavendra, and then back to Clara.
Hello, I'm Raghavendra Kadakkar from the Indian Institute of Science, from the original home of the rhesus macaque. Thank you. I enjoyed this very much. All of it was really new to me. I have two questions. Maybe I'll ask them one at a time. So my first question is, throughout your talk, I was imagining that the effect of the hurricane is quite different from the effect of social interaction and social stress on aging and immunology. But towards the end of your talk, you had one slide in which you mentioned that the hurricane actually had effects on social networks and so on. So my question is, I would expect that the hurricane would also have had effects on aging and immunity independent of its effects on social networking. So my question is it possible to disentangle the direct, if you like, direct effect of hurricane on aging and immune function, and that through social networking? Is it possible to disentangle these two?
That's a really killer question. That's a really, really great question, because, you know, I sort of slipped that in actually in one of the papers that I sent in that I didn't touch on, because I think Lauren's going to touch on it in a few weeks. It was led by Aaron Syracuse. Also talks about some of these, even though I set up these animal models as great ways to try and disentangle these different effects, there are still some of these changes that are correlated that it's a lot harder for us to pull them apart. So, right. So in the paper that I sent you, the paper led by Aaron Syracuse, we see that as we age, our social networks also change, and we're actively changing our social networks, or actually narrowing our social networks. So how do we disentangle the effect of age and age-associated changes in our social networks, and how social variation at a given age might impact the aging process, might impact our biological age, let's say. Similar question to the hurricane. If the hurricane is also altering social behaviors, you know, is that a cause, consequence, or correlate, and how do we disentangle those things? More data. Right now we haven't been we're working on that right now, but I think, you know, first we want to establish some of these ground truths, but it might be the case that some of these changes that we saw after the hurricane are driven by social environmental changes, and others may be driven by the direct effects of the hurricane, and how those differentially impact processes associated with aging in the immune system. And also we're looking across different tissues in animals that we've been able to sample, might be different. And that's a great, I say it's a killer question, because it's something that we've been spending a few years looking into, and hopefully we'll have some answers to in the future.
Thank you. My second question is somewhat more fanciful. Did anyone do a control study of humans before and after the hurricane in Puerto Rico?
So that's one of the downsides of the work in humans, and more difficulties of the work in humans, is that we often don't have the baseline samples that we want, but we do know that in response to Hurricane Maria, and also there's been some work in Hurricane Katrina back in 2006, and there's also been some work on Hurricane Maria more recently, that there are, once we look at sort of longer term consequences afterwards, we have more time, like more window of time to look at to see what had happened to these individuals. We do see increases in age-related diseases in those individuals who were exposed to it versus not. One of the complications that I sort of hinted at in studying humans, that these monkeys had to stay on the island. They couldn't go anywhere. They all lived, right? Almost all of them lived, and they had to stay there, and they all had to get the same resources afterwards. In Puerto Rico, I think there was a recent paper, maybe in the last year or something like that. There was selective immigration after hurricane, emigration, E-M-I-gration, after the hurricane, and about 500,000 to 1 million people left the island. Like, you know, like something like 10 to 25% of the population left, and so that complicates some of these analyses pre and post hurricane as well.
Yeah, thank you.
For sure.
All right, next up, we are back to Clara.
Oh, okay. When you talked about stress, it just rang a bell. In population ecology and in ecosystem ecology, stress is defined as an event that has a deleterious effect on fitness. So you could actually measure, this is a naive question or comment, you could actually measure the effect of the event on interbirth intervals, couldn't you?
Yes, so they're at the level of sort of the fitness consequences, right? You're saying here? Yeah. So there is, we've started to look at that. I mean, the Hurricane Maria was such a strong event, and these animals only get to live through one or two of them, given their average lifespan. So, and we don't have as much detailed data from before. Things that we do have detailed data on are those demographic things like interbirth interval. And we think of these as some of these things as, you know, sort of birth rates and death rates and maturation rates as vital rates in demography, and also in ecology. But when you're studying people, it's called demography usually. They're demographers. And Raisa Hernandez Pachenko, who's at Cal State Long Beach, has been doing a lot of work on Cayo Santiago and had a paper come out last year showing changes in vital rates, including birth rates, in individuals that had lived through different hurricanes, the 3 different hurricanes over the past three to 4 decades in Puerto Rico. And I encourage you to try and find that paper. If not, send me a message and I can send it to you. But one way in which you could sort of identify whether or not this was a stressor, depending on how you want to define stressor, right? If you use an ecological terminology, then at the level of like how this acute event detrimentally impacted fitness. But then we could get into a whole thing about, you know, is IBI predictive of long-term fitness in these animals? And it's like, ish.
Okay, great. And now I have some questions from Kay Sendur, a high school student from Bengaluru, India. They have 3 questions. The first one, can you comment on how the hurricane stress-induced acceleration of aging would vary across organisms with basal metabolic rates significantly higher or lower than rhesus monkeys? Would the immune response dampen or accelerate in certain animals?
First of all, thank you for, you know, I'm glad there are high school students who are here from across the world at this talk. That's really phenomenal. So thanks for coming, and thanks for asking a question, because I think it's also often the case that many people come on and don't want to ask a question. But that's a really interesting question and not something that I've thought about. So I sort of stammered a bit there because I don't know how to answer it. One thought I might have is that if individuals have higher or lower, faster or slower basal metabolic rates, then generally their life histories are a little bit faster or slower as well. And one of the thoughts, you know, we might have is that the effects of these intense stressors might be proportional to the organism's lifespan in terms of how they impact their aging rate. So you can think about something that is a bit shorter lived than macaques, then maybe you would see a shorter aging, less of an aging effect potentially, you know, than 2 years, but something that's proportional to 2 years to their lifespan. That 2 years is to macaques. We got 3 to get through, right?
Mm-hmm. Yeah. The next one you've kind of already addressed. Does the immune response vary between rhesus monkeys who experienced their first hurricane event with Maria and other older individuals who may have survived multiple hurricanes?
Yeah. So I teased at that. I didn't really get it, but since Hurricane Fiona hit last year, it was weaker, but still, you know, not good for them. And there was a big storm surge that still hit the island, not as big as Hurricane Maria. That's actually something we're looking into right now, in terms of like what do multiple hits look like, and how do individuals having shifted their environment, shifting their social environments in response to one, how does it impact how they respond to the second one? It's only been 6 months since that second one where we actually have biological data, so it's going to take us a little bit more time to be able to answer that question. Another and related question to that is actually how does not only sort of like you living through them as adults or even as like infants, but what about if you were, you know, in utero during the hurricane? And so we've got a number of females, not just females and males who were in utero during Hurricane Maria who are now adults. And actually we have a handful of females who were in utero during Hurricane Maria and actually have had offspring themselves. And we can look at some of these change generational effects as well.
Very interesting. And now finally, regarding some of the earlier parts of your talk, how would these social hierarchies vary between geographical species complex like ring species?
Oh, I mean, the social hierarchies that we're talking about there, I guess.
Yeah.
Yeah, they vary a lot, and a lot of it's due to phylogenetic inertia. A lot of these, there are lots of different theories that sort of pattern, that sort of attempt to describe, at least in primates more specifically, why there are, when and why there are strict linear dominance hierarchies, and also in which sex, males or females, you would expect those hierarchies to be, and what is the I don't know, the etiology or the underlying structure of those hierarchies. Are they matrilineally inherited, or are they more competition based? Things like that in terms of the stability or the instability of those. So a cop-out answer is that they vary a lot.
Great, thank you. And next we are back to Karsten.
Yes, just as there were no other questions in the chat, one short comment that also refers to what the students ask with repeated hurricanes. When I saw your data on the effect of the hurricane on this kind of aging, it reminded me a lot of the reactive scope concept of Michael Romero, and his wear and tear that is influenced by this. I do not know whether you're familiar with his work and also his study on the marine iguanas. Fasting. I just want to say that for me that looked very, very similar to his concept, and that would also be interesting to apply if you get more measures for future hurricanes to see whether they cope better with it, or which he would predict from the reactive scope would actually, because they already had this reactive, these costs would cope worse. Do you know the reactive scope?
I do know broadly of it, but I haven't looked into it in as much detail, and that'd be interesting to look into. If you have any articles you want to share, I just pulled this up as well, so I'll read into a little bit more.
Yeah, it's just reminded me because I have it in my teaching, and your empirical study even very nicely fits this model. So.
Great, awesome. If we have no more questions, let's give one final round of thanks. Oop, all right, we got another question from Linda Lani.
Okay, hi everyone. My name is Snyder-Mackler N, and I'm a PhD student at the University of the Witwatersrand in South Africa, working on the bush correlate, where we're trying to understand the costs and benefits of solitary living using the bush correlate as a model. So my question will be related to solitary living, because I see in your model species, they are mostly group living, and yet they are faced with all of these different costs. So would it be a way for them to try and avoid this cost by becoming more solitary living, or have you studied a similar, or maybe another related model that was solitary living and maybe living in the same environment?
Yeah, Linda Lani, that's a great question. What does the social environment mean to solitary living organisms? Because in those organisms, social factors might be stressful to them, right? And you pointed out that maybe they would become more solitary, right, and more isolated in some way, because that's more a way for them to avoid potentially more conflict or something like that. I don't know. I think that's a good hypothesis, something to test. Selfishly, I'm focused on social organisms, just sort of that's where I am. And then also because humans are social organisms, we're social organisms. And I'm interested in finding ways to understand, the consequences of our social environment, where social environment is generally more beneficial, things like that. But that's a really, really good point. I'm looking forward to reading some of your work if you're going to answer that. Cool.
Great. Okay, all right. Thanks again, Dr. Snyder-Mackler, for an excellent talk. And now we will see if we can stop recording.
Yeah.